Today discuss about structure of Embryosac. Structure of Embryosac. In angiosperms, the female gametophyte is called Embryosac. The female gametophyte is called Embryosac. The most common type of Embryosac is 7-celled and 8-nucleated or monosporic. 7-celled and 8-nucleated or monosporic. Embryosac contain three parts. Three parts. First one, egg apparatus. Egg apparatus. Second one, central cell. Central cell. And third one, antipodals. Antipodals. Egg apparatus, central cell, and antipodals. Female gametophyte is called Embryosac. Embryosac contain three parts: egg apparatus, central cell, and antipodals. Now discuss about first egg apparatus. Egg apparatus. 3 cells. 1, 2, 3. 3 cells which are grouped together. 3 cells which are grouped together at the micropylar end. At the micropylar end is called egg apparatus. 3 cells which are grouped together at the micropylar end is called egg apparatus. It's called egg apparatus. They are 1 egg cell and 2 synergids. 1 egg cell and 2 synergids. First one is 1 egg cell. Egg cell. Egg cell. The middle larger cell in egg apparatus. The middle larger cell in egg apparatus is called egg cell. The middle larger cell in egg apparatus is called egg cell. Egg cell is identified as present in basal nucleus. Basal nucleus. Basal nucleus and upper vacuole. Upper vacuole. Basal nucleus and upper vacuole. And second one is synergids. Synergids. The two lateral cells is presented in egg apparatus. The two lateral cells present in egg apparatus is called synergids. Synergids are identified as presented in basal vacuole and per nucleus per nucleus. Synergids are considered short-lived cells. This short-lived cells. Short-lived cells. Short-lived cells. That means first synergid cell is degenerated with the entry of pollen tube into embryosac, and second synergid cell is degenerated entry pollen tube into a embryosac. That's why short-lived cells. Synergids are short-lived cells. And next one is synergids have special cellular finger-like. Finger-like. Synergids have special cellular finger-like thickness at the micropylar end is called filiform apparatus. Filiform apparatus. Filiform apparatus. Special cellular. Special cellular finger-like. Filiform apparatus. Next, role of synergids. Role of synergids. Role of synergids. They nourish the female gametophyte by absorbing nutrients from surrounding nucellus. From surrounding nucellus. Absorb nutrients from surrounding nucellus. Next, the filiform apparatus of synergids. The filiform apparatus of synergids secrete chemical substance which help in the guiding of pollen tube into synergids. Pollen tube into synergids. And next one is central cell. Central cell. Central cell. The largest cell in embryosac. The largest cell in embryosac. The largest cell in embryosac, which contain two polar nuclei. Is called central cell. The largest cell in embryosac, which contain two polar nuclei, is called central cell, which fuse to form a single diploid, a secondary nucleus. Largest cell in embryo sac is called central cell. And last one is antipodals. Last one is antipodals. Antipodals. Three cells which are grouped together. Three cells which are grouped together at the chalazial end is called antipodals. Antipodals are smallest cells. Are smallest cells. Are smallest cells. Antipodals is also called as vegetative cells. Vegetative cells. Vegetative cells. Three cells which are grouped together at the chalazial end is called antipodals or smallest cells or vegetative cells. These three cells degenerate before or after fertilization. Before or after fertilization. Fertilization. The largest cell in embryo sac, the largest cell in embryo sac, that is central cell. Largest cell, central cell in embryo sac. The smallest cell, in embryo sac, that is antipodals. This is structure of embryo sac. Thank you.